Good day, my friends. Welcome back to the channel. Today we're going to discuss cultivation of viruses. And the question to ask now, wetin be the methods wey we dey use cultivate virus? If we wan prepare virus, if we wan make many copies of virus, wetin be some of the ways wey we dey use cultivate virus? These na the questions wey we wan answer for this video. Now, e dey important to note say a fully matured virus particle wey get capsid all around am, sometimes we dey refer to am as viron. This one dey important to note. Now, e get many methods wey we dey use cultivate viruses. Three major methods dey. One na wetin we dey call animal inoculation, meaning say we go put that virus inside animal. The virus go come dey multiply for within that animal. A second method na say we fit put the virus for inside wetin we dey call embryonated egg. So egg of chicken wey get embryo, we fit put the virus inside make e dey grow inside that egg. Now, a third way wey we fit use cultivate viruses na to put that virus for inside wetin we dey call cell or tissue culture. This na one of the three ways through which we dey take cultivate viruses, and this one dey very important to know. Also, we don talk am for our former video say virus na wetin we dey call obligate intracellular parasite. This one mean say number one, obligate, meaning say e no fit survive outside living things, intracellular. So e need host, e need living cell wey e go invade, come dey function, come dey replicate for inside that living cell. So this one dey very important to note. Now, the question to also ask na say wetin be why we dey take cultivate viruses? Wetin be the reasons why we dey take cultivate viruses? Now, some of the reasons na number one, if we cultivate virus, we dey fit identify the virus. E go help us identify virus wey dey for inside any clinical sample. Person dey sick now, come hospital, dey fit take im spit, dey fit take im urine, dey fit take im feces, dey fit take im blood. Anything dey fit take for im body, come take am, carry am go lab, run tests. So to study viruses dey help us to dey fit identify viruses wey we fit find for any of these samples wey we dey call clinical samples wey we fit take from the body of person wey dey sick. Another reason why we dey take cultivate viruses na say e dey help us for research. If we wan understand the structure, the function of different viruses, na only if we fit produce many of these viruses na we go fit do research on top these viruses. Also, e dey very important to prepare viruses for vaccines. So we dey cultivate viruses so that we dey fit study these viruses, and so that if we wan produce vaccines, we fit use these viruses wey we don cultivate, we go use am produce many different vaccines. We know say many vaccines dey for viruses, such as COVID-19 wey just finish, wey just pass now. You know say vaccines dey for all these viruses. Now, like I talked before, e get three major methods wey we dey use cultivate our viruses. We no dey use culture media like bacteria. These three methods na either through animal inoculation, through we go put am for embryonated egg, or we go do cell or tissue culture. Now, for this video, we will put our mind down, discuss animal inoculation as one of the methods. We will do other videos where we will talk about other methods wey we dey take cultivate viruses. But for now, we'll talk about animal inoculation. And wetin inoculation mean? Inoculation na big word for science wey mean to inject something into something, or to take something, take a sample, come put am for somewhere make e dey grow. Na wetin inoculation be. So animal inoculation na method wey be say we will take virus, come inject am inside animal, make the virus dey grow for within the animal, dey replicate, so that we will get many copies of the virus after a while. Now, which animals we dey use for this inoculation? We fit use many primates, chimpanzees, monkeys. We fit use rabbits, we fit use mice, many different types of animals. But majorly for science, we dey use mice most of the time. And that mice na usually mice wey dey about 48 hours old. So no be mice wey don old, na mice wey still dey young. And the reason for this na say virus dey replicate better for any organism or cell wey dey divide rapidly, wey dey divide fast. So that na why e dey very advisable to use 48 hours old mice to do animal inoculation. And many a times we dey make sure say that mice dey very free of disease or illness. Now, e get many routes, many methods wey we dey take inoculate that mice with the viruses. Some of these methods include one, intranasal. Nasal na for nostril. So we know say intranasal mean we go make that mice make e inhale that virus. If you don inhale that virus enter im body, the virus will come dey replicate for inside the body of the mice. That one na intranasal. The second one na intracerebral, meaning say we go inject or inoculate the virus into the mice through the brain, through the cerebrum, the brain. So we'll put am there. So that one na intracerebral route. Now we get subcutaneous route. Subcutaneous route na through the skin. So if we inject the virus through the skin of the mice, make it enter the body of the mice, come dey replicate, dey divide for there. That one na subcutaneous route. The last route wey we will discuss na intraperitoneal route. E get wetin we dey call peritoneum, and peritoneum na wetin we dey refer to as body cavity. Inside every organism, e get something wey be like hole wey dey for inside our body. We dey call am peritoneum or cavity. So sometimes scientists dey take the virus come inject am deep into the peritoneal cavity of the animal. If we do that kind of inoculation, we dey refer to am as intraperitoneal inoculation. So these routes dey important, and na the routes wey we dey use to inoculate different animals with our viruses. Now, wetin be the advantage of animal inoculation, and wetin come be the disadvantage of animal inoculation? The advantage of animal inoculation na say e dey easy to do, e dey easy to use if you wan diagnose of any viral infection. E also dey easy to use as a method if you wan dey study pathogenesis, meaning we wan study the way wey viruses dey use cause infection. E dey easy to use animal inoculation to understand am. Now, e also dey easy because a lot of antibodies dey produced very quickly by the mice. So e dey easy to harvest these antibodies and to fit study all the structures and functions of the viruses very easily. And also, these viruses dey help in the study of oncogenesis. Oncogenesis na the study of cancer, the way wey cancer dey take form. E dey easy to study cancer and information in animal inoculation when you inoculate animal with virus. This one dey very easy to understand. Now, wetin be some of the disadvantages? The disadvantages na say e dey expensive. Mice dey expensive, and you go dey buy many if you wan dey run this animal inoculation. Also, many a times we realize say e dey difficult to keep the mice, to take care of them, to feed them, to all these things na processes wey dey difficult and dey laborious. Dey spend time, dey time wasting. So these na some of the advantages, some of the disadvantages of animal inoculation. Another important disadvantage wey we go use take close na say some viruses, if you inoculate them into animals, one, they no go grow, or secondly, they fit grow make they no cause any disease, make e dey difficult to harvest them out of the animals. This one too dey happen. Sometimes, so a disadvantage na say many a times e dey difficult to determine which animal we go take to use to grow which virus. How we go take match them, know the animal wey go grow inside, be the virus wey go grow inside which animal. These na some of the disadvantages wey dey for animal inoculation. We will do some subsequent videos where we will discuss all the other types of methods wey we dey take cultivate viruses. Thank you for listening to this our video. Abeg, make una like our content, make una share our content, make una friends dey see am. The whole idea na just to make our science better. Have a wonderful day.